What did you say, 295?
Yeah, 290.
290.
Bridge knock.
Give me a sense of how steep it is. Yeah. Turn around to.
At step 100 meters, bearing 290. Thank you.
The cloud that means there was a fish there.
Yeah, it might have been my lateral thrusters as I turned there.
Another Munida.
Reporting head. Oh, we got him.
I was gonna say something flat and striped.
Yeah. Stripes throwing me off a little bit, but it's probably a Dover sole.
Quick zoom there, Steve. So we come out. Thank you.
Low, longer, like 12 fin. Could be a Rex.
I was gonna say that looks like a Rex fin.
Yep. To me, it looks like a Rex sole.
That's the one I recognize because of the fin.
These and top tillum sea pans always look like question marks to me.
Mm-hmm. They were calling them squirrel tails earlier, which I thought was really apt.
Yeah.
Yes. If only they'd be able to kind of flutter it like a squirrel could every once in a while.
Yeah, no, there's that hook.
Definitely patchy habitats here. We'll get to some more mud and a few rocks, and then we'll get into higher density rocks and boulders and go back to this.
I think it was
Yeah,
just bio.
Yeah, I think so.
Still.
Yeah, it's like corner of the screen, so I couldn't really tell.
Yeah.
What's happening there?
I was just a line-like object floated by.
Yeah.
Oh.
We're all on high alert.
Most likely, Siphonophore.
Oh, we've got a long fin fish here. Is that a black dragon?
It does look like, yeah.
Zoom in, Steve.
Pacific black
Yeah,
dragon.
turn the porch light on, please.
It's off.
Ah, too late.
Oh.
That one held still way longer than they usually do.
Yeah. Back to the comment, Jake, about the fishing line. I guess I did see some monofilament on the seafloor earlier that was wrapped up in a kelp hold fast, but I haven't heard how much they've seen throughout the whole dive.
We're on high alert. We've had 2 situations already this season. So.
Yes. One as recently as last night.
Yes.
That was a major one.
That was fun.
Definitely.
It was a very stinky one.
Yeah.
Was it?
Oh, guys, the whole back deck was full of mud and Scientists are just having a field day on a pile of rope.
Yeah, it looked like it had been down there for a while and was thoroughly encrusted.
Definitely smelled like the bottom of the ocean.
Some of the nautilus crews quite into taxidermy as well, so the hanger is rather pungent. Smells like the wet lab.
This gives a good sense of how sponges are really good at filtering water. That inside of the sponge is like perfectly clean.
That's how you can tell those ones are alive, is when they die, that inside stops being perfectly clean.
Mm.
Yeah.
It's like some kind of biological Fabergé egg.
Yeah.
I think I read in one of Henry Weisswig, sponge taxonomists off the west coast, located up in British Columbia, that unfortunately recently passed away. One of his early papers talked about sponges could filter up to 20,000 times their volume in a given day, the volume of water. Kind of an amazing amount of water.
I wonder if that is the star calyptus Pamela Turneri.
Talking about the yellow one?
Yeah, the yellow one.
Yeah.
Zoom in, Steve.
Certainly an interesting form.
Good, thanks.
That's our groove tanner crab right there as well. You can count in 4 legs.
Some interesting stuff growing on its shell.
Yeah. It's like some polychaete.
Oh, Abby's joined us. She can tell us what the sponge is.
You want to stuff him up for a minute so I get
Bird
back?
snuff. Hold position.
Okay.
Thank you.
Get a quick shot of the crab there too.
These look like
Out for
little
that rock.
worms growing on its shell.
Yeah. It's hard to tell if that crab is missing a leg on the right side, the back right.
One more
Odd
quick
number
zoom
that I
there,
can
Steve,
see.
before he disappears behind the sponge.
Hiding.
Okay.
Shy,
Off we go.
it's a shy crab.
It does look like it's missing a leg back there.
And zoom in there again, Steve.
Another one. Is that a squat in
Yeah,
the
good,
background?
Yep.
good.
Great shot.
Okay.
See, that combined with the polychaete makes me think that this crab has not molted in a while.
Right.
Do they molt on a set schedule, or is it kind of health dependent, like whenever they're ready to?
I think that might be somewhat species dependent, because I know it's somewhat, they definitely do it as a size thing. They'll just outgrow their shell, but there's definitely a season, at least up here with some of our species, where you'll get all of them kind of molting all around the same time.
a couple Dover sole, small ones on the bottom of the screen.
Mhm. How far have we come since our last transect?
Not far.
Okay.
It's about 60 meters.
All right, so we'll just keep going for a while before we.
No, there's lots of little thorny heads around here. And that's a thorny head too.
I thought I saw an
Where am I?
aurora
Slope
earlier.
ice in the middle?
Yeah, we're getting like,
Sorry, what
what
was
are we
that?
at,
Where
465
am I?
meters?
Slope ice, like in the middle?
Yeah, right here, here, but
Right.
it doesn't seem like it'll be like cliffy. Probably just a steeper version of what we're looking at. Ready to move?
Yep.
Bridge
Thank
nav. 100 meters, 290. Thank you.
Yeah, as Meredith mentioned, Aurora rockfish are one of the deeper rockfish species in this area. But thorny heads are generally deeper than rockfish, or at least they extend deeper than rockfish do. Their range does.
I don't see the thornies all the way down at 12, 1300 meters. Most of the other rockfish don't see till they get up till the 600 meters or so. They can go deeper, but they don't frequently. Mary's filled us in on the molting. It said tanner crabs, like others in the spider crab superfamily, have a terminal molt. When they reach sexual maturity, they cannot molt again and so cannot regenerate appendages or broken parts of appendages. They often become encrusted with tube-building worms, barnacles, and sponges. Thus, the missing leg and the tube worm growing on the shell.
Makes sense.
That's great information. Thanks, Mary.
To be young again and able to molt.
Hmm.
They have to give up their reckless ways once they reach maturity. Can't grow those legs back anymore.
A quick zoom there, Steve.
Almost like a talus slope here.
Good eye.
Mm-hmm.
That is a good eye, Dan.
Okay.
What's that?
I can't tell what that is. It's
Yeah,
just
I'm
this
not sure.
weird little arm sticking out of the.
Yeah, very translucent.
Say on the seafloor, just guessing polychaete is generally a pretty good guess.
That flatfish was really good. Buried, half buried, up to the right. We just passed.
It's another one of those little weight things. Zoom in there, Steve. What am I?
That looks like a worm, right?
A worm?
Worm casing.
Yeah, that's a worm.
Okay.
That's a polychaete tube.
All right. I'll go up the hill a little bit.
Roger.
Mary took a look at the crabs that were on that sponge and says that they're Corellia longipes. Also some of the larger crabs we've seen nearby. And there's some controversy over whether this is one species or two. the key to look for is between the eyes, the rostrum. In Corellia, the rostrum is deeply forked.
Oh, yeah.
And for those at home, if we zoom in on another one of those crabs, the next time we see one, the rostrum is what looks like the nose right on the front end of the crab between the eyes.
More of these yellow sponges. Was it the Staurocalyptus that we thought? Is that what we're thinking those are?
That was my guess.
They're hake down on the bottom right.
Abby says Staurocalyptus pamela turneri was described from a piece of sponge growing on the wreck of USS Independence off San Francisco.
Oh, wow.
I think we dove on that, right?
Yes.
Yes,
A few years
that's the
back.
one that had the plane
Yeah,
on the deck.
the Hellcat right on the deck. That was pretty cool.
Abby's saying a sample would be good if we're allowed to do that.
For which?
The yellow sponge that
Next.
we just pat. Yeah, next time we see one.
Yeah. The Staurocalyptus is another group where there may be more species than have been previously described. And one of the groups that every time Henry would look at them, he would say, yeah, this isn't the same thing as it was the last time.
Is this the same one that you want to sample or different?
No, it looks different.
This one's different.
Bright as yellow.
Yeah.
Yeah, this is different. This is Mike Haley. So we've seen 2 or 3 of the Staurocalyptus, so next time we see one, we'll take a piece. We seem to have found the steep again.
Yeah, we're in this bit of a steep patch. Doesn't seem to be too much of a wall, just kind of a failing slope.
Just to the left of the lasers, that might be one of the Staurocalyptus growing on another skeleton of a dead sponge.
Zoom in, Steve.
Does that look like it?
It looks like it to me. I'm waiting for Abby to chime in on the science chat.
Sure.
Yep, she says it
Says
looks
yes.
like
Bridge
it.
nav. Hold position. Thank you.
Okay, can zoom out.
I think we'll have the leash there for it to stay.
Nice little aurora rockfish sitting next to it. The lights in my office just blinked.
Zoom in there a bit, Steve. Yeah, zoom right in. And you guys want the whole thing there?
Let's see what Abby says.
I just grab the dead sponge and get that all in one if you want, or we can slurp pieces of it. Show us a
I
minute.
think she's on the building.
She says it's a grab if you can get a piece
Yeah,
off
right.
of it.
Zoom out, please. Oh, my landing rock. Game boy.
No.
And Aurora Rockfish is defending its territory.
Yeah.
I think it's going to be the law of tonnage. And Abby does say if it's the whole thing, then that's fine. It's harder to break a piece off.
If I can function this thing in flute mode.
You gonna try?
Freaking painful.
Oh,
Zoom back in a bit there, Steve. Show me the sponge. Thanks. We have samples in the front in the front box.
Yeah, but you could put it in forward box A. There's just 2 urchins in there, so the left box would be fine.
Oh, there's a
They're all
little ruby
connected
nose
there.
Bertrula.
Oh, yeah.
Just for Tom if he reviews this video.
That's a great grab.
Nice grab, yeah.
Get a quick zoom there while we open the box. It's a little too much there. Or not. Yeah, it's good. Perfect. Okay, you want to bump the box out?
See those really long spines coming out there from that sponge. Those really
Yeah.
long
Zoom out,
cutter
Steve.
guard
Thanks.
spicules.
NA, you say, huh? It was A, right, Taylor?
Yeah, A is fine.
Thank you.
What's the number there, Taylor?
Sample number 117.
117.
Abby says, great sample, thank you, and that she will be looking at whether those long spicules are thorny or smooth under the microscope once it's back in the lab.
But positively buoyant on the top there, huh? Good thing that you grabbed that dead sponge underneath and maybe part of the rock. It's holding it down.
Back out into the cobbles.
Come down 5 and look up a little, Jake.
Coming down.
Current's favorable,
Up,
the
Tony.
tether streaming off to our right, and we're moving that way too, so that's better.
Some of these shaggy barrel sponges are almost invisible.
Okay, we're moving. Can
Bridge
move.
nav. 100 meters, 290. Thank you.
That white star on the right almost looks like a mediaster. But I know Erica Burton's on. Maybe she'll correct me.
Sorry, what were you looking at?
Oh, there's just a small white star off to the right.
Do you want
Just
a
trying
closer
not to
look?
bias myself with fish. No, that's okay. Thanks, Dan.
About to get all excited about the orange thing, and then I realized it was a crab just hanging onto the rock.
Another raw rockfish, below yellow Michaeli sponge, and then here's these larger barrel sponges, which Abby did ID earlier for us. I thought they were the Rhabdoclyptus.
Yeah.
Nice pair of them, though.
Whenever they
They
have
stayed.
that like really just straight base morphology, I always think, oh, that'd be, they'd look wonderful as some kind of like, object to art. They would smell terrible, but they would look great.
A crab down in the bottom of the barrel sponge.
Yeah, I saw that in bubble cam. Somebody's home.
Reminds me, I was on a work trip in St. Petersburg and was just kind of waiting to go to the airport and walk down on the beach and one of the bridges or causeways there, and there were some horseshoe crabs and there was an old shell. So I'm like, oh, I'll take this and show it to my daughter, who was much younger at the time. And I think when I finally got home and I put it in a Ziploc bag, but I opened my suitcase like the next day after a late arrival that night, and it was a pretty intense stench that was coming out. I don't think the Ziploc could contain it.
No. We used to flip those over at the beach, Delaware. Horseshoe crabs.
It's more fun when you slip a little something something into your mate's bag when he's leaving the boat.
Yeah, right.
Grenadier tucked in behind that sponge.
I guess they can change direction when their rock falls over.
Yeah, like trees staying upright when there's
Right.
a soil creep.
near Palo Alto in the Bay Area, there's a earthquake trail that traces, some features of the 1906 earthquake, and there's a handful of oak trees like that toppled over during the earthquake and just kind of changed direction. Or like, one of the branches turned to be a new trunk, for instance, because it had tilted over 90 degrees. Another nice yellow sponge.
Yep. A little flatfish hiding underneath it.
Oh,
Yeah.
It seemed as skittish as the trolley the other night.
Oh, they were funny.
Yeah. Aurora rockfish in the middle of the screen there. Yeah, those petroli, it's almost like that's how they mask their escape. Big dust cloud, and then off they go.
Yeah, I never thought of that as
Amazing
a defensive
density.
move, but that's a good idea.
Yeah. Amazing density of fish, though.
One of those scenes where the whole bottom is moving.
Yeah. More of these media squat lobsters.
Those arms seem to be getting longer.
Is that, what are those white things right there, man? Is that a paragorgid?
Zoom in, Steve.
That's those club sponges. I
Oh,
think.
okay.
False paracord.
Oh, yeah. It's like antlers.
Mm-hmm. The yellow color that gives it away.
Yeah. That's a nice thorny head picture.
Roger.
You want me to slow up a little bit? Oh no, actually you're kind of in line.
Try and make up a little ground here. Lives in the battle.
Also,
Tony,
slow to
it's
point
sure
two.
like it down here.
Sure.
Mm-hmm.
Bridge now.
Oh.
Slow speed to 0.2. Thank you.
Oh, there's another flatfish almost totally buried. Just to the right of the lasers.
Oh, yeah, to the right of the lasers, yeah.
Zoom in real quick, Steve. That's good, thanks.
Good camo.
Really good.
Okay.
Nice shot. It's good to oversoul. And this one in the bottom of the screen is just kind of draped over a rock.
Seems to be a lot here.
Yeah.
Oh, and another header coney.
Yeah, I wouldn't have called this typical flat. Sorry, go ahead.
It's a nice peach color.
It is. Wonder if there's anything hiding inside.
I always wonder the same thing. Not quite that. I can look at one at our down-looking
snap.
camera. Not quite the right angle for the peak. Oh, maybe.
Hold position there. Thank you.
Oh no! Oh no
There's the angle.
no!
Oh,
Angle
See in
adjustment.
channel 3, you can see our
Zoom
downward camera.
in there, Steve. Thanks.
pretty clean in there. Definitely attached to a dead
Nobody
sponge,
home.
though.
Okay, zoom out. Thanks.
I just stopped up for a brief minute.
Yeah, Reg. I don't know how I got so far behind. It's a turn and go.
Yeah, I'll keep going at 0.2. It seems like that's the pace.
So much to see after days of mud.
Yeah.
Lots of
Gives you a sense of
polycube
the
tubes
slope
on these
there.
rocks. Oh, wow. That's great. Great shot. It is. It's like a talus slope.
It's pretty steep.
Rocks, it's
Yeah, it's nice
raining
to see
down
that
from
perspective
above.
you lose. Go ahead, Meredith.
I said, rocks, it's raining down from above.
Mm-hmm. On that dive, can't remember how many days ago it was, but on the Palm in San Lucia Bank, we had some talus slopes that were going over there, but I don't think we saw very few sponges on those. So almost makes you wonder if that was more recent movement of those rocks, or if there's some environmental conditions that just didn't support sponges to settle in those areas.
Even at the start of this watch, on the talus slope we started on, there weren't quite this many sponges.
Hagfish
Little
coiled up.
hagfish at the bottom.
Yeah.
Oh yeah, it's like a cinnamon roll. Although
The
a lot
world's
slimier.
least tasty.
Causing an avalanche.
So Abby commented in the chat that what we might be calling heterocone could be a different species, Pinelasma.
Zoom
Can you
there,
zoom in
Steve.
on this?
Is that? It almost looks like a different type of sponge growing on the store clip. It's like a fairy almost. No, it's just different color. Interesting.
Sponge on a sponge.
Yeah. Or it's lost its color or something. It's really strange.
Yeah. It looks like it's a slightly different texture too.
It does. And more of a Mycale type sponge below it too. See if Abby gives us some clues.
I imagine if it's a different species, that can be one of those. nightmare to tease out scenarios where you're like, well, which spicules are from which sponge?
Yeah, right. Almost looks like there's something inside it too. Maybe a little, could be hydroids, could be a brittle star. It's hard to tell. Abby kind of confirmed what we thought. Yellow star calyptus with a different glass sponge growing on it and a mycale at the base.
Everybody's saying that the bright yellow staros might end up being a species complex, so there's likely to be more than one species in that group.
Yep, that we won't be able to tell just by looking at it through a video feed.
I can see a mushroom coral looming in the background there, as much as a tiny 4-inch coral can loom.
Those are great shots of those sponges, though. Thanks.
My pleasure. Moving on.
Bridge now. $100 meters, 290.
Come down 5.
Coming down.
Please.
Another hagfish coiled up there in the bottom left screen.
Oh yeah, I see him there.
Oh, that sponge really blends in, doesn't it?
Oh yeah,
sediment.
almost invisible.
Quick zoom. Quick gross out zoom on the hagfish.
Oh, it's just reared its head.
I give you the full taste experience.
Wrinkly. Gives you a good shot of the mouth, though.
Looks almost leathery. It's weird.
Yeah.
So there's actually a fishery for these guys for their skin.
Really?
If you see things that are sold as eel skin, it's often actually hagfish.
Okay, moving on.
Yeah, that's enough of that.
Yeah, I don't know. Meredith, and maybe you remember, but several years ago there was they were transporting a load of hagfish on a semi-trailer
Yeah,
on a highway in Oregon, and it rolled over and dumped all these hagfish and slime all over the highway, and it was a really messy cleanup.
Those some great pictures.
Slimy.
State trooper, his car and him all just completely slimed.
There's that mushroom coral, Meredith, you saw on the bottom of the screen.
Yep. So far, I've only seen the light color morphs of the mushroom corals here.
I'm not seeing it.
Hey, it's just bottom right, just off screen, so it's a little bit below you, Dan.
I dust it.
Yeah, probably. It's all right. Yeah, probably a little bit to the right.
Yeah, I got too close there.
There you go. It's that white thing on the far right of the view now.
All right.
Cool. white disc.
It's got all its polyps tucked in, it looks like.
Guess he didn't like dust. Zoom in there, Steve.
Reminds me of watching a Smurfs cartoon growing up.
When they get all the
color,
polyps tucked in, get all the polyps tucked in and they're up on that stalk, you can really see why they're mushroom corals.
You can go back for a tight zoom if you want. It's cleared up a bit.
Oh, that's okay. We can keep going. Started to see a few more of them mixed in, so I'm guessing we'll get another opportunity.
Yeah, hopefully.
Yep, definitely seeing more of these Storoclyptus yellow sponges. It's good to see.
How far have we come from the last transect now?
We are 200 meters from the last
All right, we
transect.
could go ahead and start another transect.
Zoom in there, Steve.
Oh, yeah, let's look at the sponge first.
Yeah. There's a mushroom coral right above it with the polyps out this time.
Coral. And that
Dan was
there is
determined
the
to find
sponge
us
we've been
one.
calling the white sparkly sponge.
It's pretty. Does Abby know what it is, Meredith?
We have no idea what it is.
Zoom in a bit more if you want.
We sampled one last year, and if we see another one, now that we've seen one, we may sample one.
Okay. Great shot.
Yeah, we saw it. I've seen it in the AUV imagery, and I've seen it. We've actually encountered it now a couple times in ROV imagery, but we have no idea what it is.
Pretty cool.
Looks like there's a feather duster worm coming out through it too.
Yeah.
The bottom there.
This one is interesting because when it's on the surface, it stays really rigid and it stays this kind of sparkly looking.
Is it
I'm
kind of
chatting.
You've seen it? You've seen it on the surface, Meredith?
Yeah. So during Express last year, we sampled one from a little further north.
Okay. Does it have kind of like a fiberglass texture?
Yeah, kind of a crunchy fiberglass texture.
Okay. I know our trawl survey gets a sponge that they describe as fiberglass, and it's really, when they get a larger clump of it, it can be really really tough to process, to get it out of the net.
Yeah. I don't think this is that one, but I would have to ask JB.
Yeah.
You want me to hold here?
Yeah.
Great photos.
Great snap.
Lots of hydroids
Tilt up and
covering
look at-
that it's a rock or another sponge. It's hard to tell. Well, we'll have to keep an eye out for that for another one, right?
Yep. We can go ahead and zoom out and get set up for a transect.
And you want a tight shot on the mushroom
Sure, let's take a quick shot on the mushroom.
while we're here?
It's looking particularly photogenic.
Totally.
All the polyps out.
Yeah, that's a great shot too.
Those have such a super weird texture on the surface. Almost like softball-like. Simultaneously kind of rubbery and crispy.
Wow. This is a good shot showing why these are called octocorals.
Mm-hmm.
Oh, that was cool to see that one close.
And these guys can pull these feeding polyps all the way in, so and then kind of close up the calyx around them, so you get almost a smooth surface, just with little
It'd be great to see a
dimples
time
everywhere.
lapse. Yeah, it'd be great to see a time lapse of this, pulling them in.
Yeah.
Looks like one of those polyps only has 6 arms.
Oh, yep.
Oh yeah, it is missing.
It lost them to
I can't see
something.
the nubs.
There's a variety of different sea stars, nudibranchs, amphipods that will feed on these corals. And this group, the Heteropolypus, there are 2 color morphs. One that's just a solid dark pink and the polyps are the dark pink. And then this light morph with the darker pink polyps. And as far as we've been able to tell so far, they are a single species, although we've only done mitochondrial sequencing so far. And there are some octocorals that across multiple species can have identical genetic mitochondrial sequence for the genes we looked at.
Yeah, that's a really interesting, you're kind of at the forefront of it, Meredith, thinking about how genetics and traditional morphological taxonomy intersect.
Yeah, I have a feeling in my future on some of these groups that we're looking at where we're starting to find cryptic species, we have a lot of putting together kind of charts that have individual nucleotide changes paired with, it's got pink polyps, it's got yellow polyps, it has this type of sterite to get it all kind of into one space of what you're looking for. Because on some species like this, color doesn't seem to be a big indicator, but on some of like the sea pens, it can be.
Yeah.
All right, I think we can zoom out and get moving.
Okay.
Yeah, thanks for those zooms.
Our pleasure.
I'll let you get ahead and then I'll do 0.3290, do a transect.
090?
290.
290, roger.
Yep.
Flatfish is almost center screen, a little bit to the right now. Okay, this view gives you a nice idea of how steep the slope is here.
Yeah, we flattened out for a while, but we seem to be back in the steep for a while now.
Mind if I go for a little zoom on Argus?
Yeah, I don't mind. Bring it, Argus head to the left a little.
It's a good look at the lateral thruster that you don't usually see.
Is all the Niskins lined up?
Another nice barrel sponge right in the middle there, and then a Mycali like yellow sponge on that rock, and then looks like an Aurora rockfish to the right.
Nice little marine tableau.
Yes. A couple of small barrel sponges on that rock to the kind of bottom right. Barely see it.
Okay, you ready for me to start?
I think so, yeah.
I'm wide on Argus again.
Bridge now.
Roger.
200-meter step, bearing 290, speed 0.3. Thank
Tell
you.
me that's a siphonophore.
The one bouncing from the top.
Yeah.
Yeah, thank you.
Oof.
There's been a
You're
lot
gonna
on this
be-
dive.
Yeah.
You're going to be the most hated of the pilots'
Yeah.
animals.
At least the seagrass stays on the seafloor.
Frightening things. Am I down at the bottom of the slope again or in the middle?
You're in the middle,
Roger.
but it'll eventually peter out. Hopefully we'll have this slope for most of the transect.
We're doing a sideways transect mode, are we? Is that what we're setting?
Roger.
Okay.
Yeah, that looks good, Dan.
Yeah.
Check the camera here for lighting. It's a little high on the last one. All right. Oh, he's going already.
Volcano
Is it?
crab on the
Yeah.
right.